Okay, we're continuing with the last little bit. Here, our essential question is, what are the cell's structures? And their functions. So the functions of those particular structures. So we've got a few more kind of categories to cover, and then I'll show you some little review bits that I have for you for the chapter, because there's a lot here, and a lot of this stuff is important. They love to ask questions on this on STAR, so use the class time to ask questions if you need more things. I've got resources all over the place. All right, but some things we need to know first are the mitochondria. We're going to spend a good portion of time reviewing mitochondria and their structures and stuff like that in Unit 3. But these are the organelles that carry out cellular respiration. So cellular respiration happens in all eukaryotic cells, or most of them, if you will. But actually converts chemical energy in foods to chemical energy that we use in the cells called ATP. It's like a little AA battery that just happens to run absolutely everything in your body. It's a small molecule and gets everything done. It's really impressive. So is the how-to on that one. That's why it gets its own unit. Nonetheless, here's mitochondria and what they look like. They always have this kind of shape where they've got these little folds and wrinkles in them. And I'm not doing it justice, but you see kind of what it looks like in a 3D shape. We'll get more into these particular parts as we need them. But the big idea here is to recognize the mitochondria, what it looks like, and what it does. So photosynthesis is the conversion of light energy from the sun to chemical energy of sugar. So photosynthesis will take light energy and make sugar. So chloroplasts are the things that do that, and they're the photosynthesizing organelles of plants and algae. You may recognize algae in pools or even fish tanks. It's that green stuff that grows. So because it's green, it's got some relationship to plants. All right, they have a different kind of look to them, and we'll get into these. We will know these by the end, but you would be able to tell it because they usually represent it green. It's kind of got two layers to it, and then there's these little coin stacks in it. Okay, so we'll get to know these in more detail next unit. So here's a little connection for you. May ask you just a teeny bit on this one, but it's fascinating. So mitochondria and chloroplasts actually contain DNA and ribosomes. They contain their own stuff. So the structure of DNA and ribosomes are very similar to those found in prokaryotic cells. In fact, you guys get, so the DNA that you would find in your own mitochondria in your cells comes from your mother. It's maternally transferred or inherited, I guess is better. So you're always carrying your mom's mitochondria. And that's kind of the way that works. So the idea, because it has its own genetic output that's completely different from everything else, is they kind of started to think about this endo-symbiont theory. So it sounds like a big word, but endo just means within. So within. Sim is together. Just put all the words together. Bear with me on this one. Bio is life. So this is a theory where life came together within. You're like, what? Okay, so what it says is that mitochondria and chloroplasts were formerly small prokaryotes on their own, but they began living with larger cells. Basically, like, here's a little mitochondria, and then at some point, a bigger cell came along and ate it. It's like a Pac-Man cell. And then eventually it became a cell that, had a mitochondria in it. He's still Pac-Man. There you go. Okay, so here's sort of what we would have had is like maybe a nucleus and some endoplasmic reticulum, so more of a complicated prokaryote forming. And then it would have sort of engulfed or eaten these mitochondria and taken them in. So these would have formed our non-photosynthetic eukaryotes, something like us, right? And then a little bit later on, maybe something else engulfed this prokaryotic, like photosynthetic prokaryote, and then we started to get these photosynthetic eukaryotes, so plants and stuff like that. That's the idea. So you kind of see what's happening here, is that we started off with one cell and it began engulfing these smaller ones. I don't know, but it's a fascinating thing that we'll look at. So the cytoskeleton, again, cyto is just cell, so this is the cell's skeleton. Okay, and we're going to talk about it briefly. It's not one that's super discussed all that much as far as like STAR goes, but it is at least worth knowing if you're going to move forward in biology. Even if you're not, it's handy to know. All right, so cells contain a network of protein fibers. So proteins coming up again. That's why we had to learn some about those macromolecules. And that network of protein fibers is what makes up the cytoskeleton. So they organize the structures and activities of the cell. They're very organized, and they form even a highway system. Let's actually do that. So just so you kind of know, they organize structures, so they're going to be sort of highway-like because they can transport. Let's get that here too, is that they'll do some transport. Okay. They'll also kind of serve as an anchor, so they'll make sure that cell parts kind of stay in places. After looking at your own cells, and some of them will be folded over or whatever, but the idea is that if you see the nucleus, it's not like stuck over to the side at one point, it's not, randomly at another. It should be in the center a lot of the time, or at least close to it, and that anchoring comes from the cytoskeleton. So those are some sort of big functions of that cytoskeleton. So we'll go into a little bit more detail. Again, don't have to know a whole lot here, but there's a couple of them. So we've got microtubules. They give that shape and support of the cell, and they act as tracks along which organelles are equipped with motor proteins to move. I gotta find you a video. It's the cutest thing ever of a little motor protein walking around on a microtubule just doing his little business. It's the cutest thing. He's got his little steps, and he's got a little pep in his step. It's pretty cute. I don't know, what's the term? You guys gotta tell me what the term is that. They don't have swagger. That would be weird. I don't know. Come up with a better term for just like pep in his step. Whatever vernacular you kids are using these days. Just messing with you. Alright, so in animal cells, microtubules grow out of a region called the centrosome of the nucleus. So again, this is only animals. So we'll see the centrosome and we'll see these pair of centrioles only in animal cells. So this is one of their key differences. And it's made out of these microtubules. We do have some intermediate filaments. So these are found in cells of most animals. They reinforce cell shape and anchor those organelles. Okay, and they're often are really permanent fixtures of the cell, so they just kind of stay where they are. So again, anchoring those organelles, keeping them in place, making sure that cells have a nice shape and then don't get deflated for no reason. No, giving them a nice round shape. We want things to be the way that they're supposed to be. Microfilaments, these also help support the shape and actually are involved in motility, so movement. That's where we start to see those actin fibers. So it's involved in motility. So the big thing from the cytoskeleton, if we can know that it's involved in anchoring, support, and sometimes movement, I forgot that in my first one, that's a good thing to add. Okay, so you can sort of see, they've got some really good pictures here of what's going on, but a microtubule is see the little fuzzy stuff going on in there. Intermediate's a little bit more of the fuzzy stuff, keeping stuff together. And then some of those actin fibers for movement. They're small, but we'll see those again later. And if not, you will see them in anatomy when you get to the muscle section. Here's just a really cool thing, talking about actin and myosin in general anyway. You don't have to write any of this down, but it's a cool history thing to notice that in the 40s we had some biochemists who isolated and identified proteins, actin and myosin from muscle cells. In 54, scientists usually are using some newly developed techniques from microscopy established how filaments of actin and myosin interact with muscle contraction. So for the next decade, they researched the actin filaments in various types of cells. In the 70s, they were actually able to visualize it using some cool fluorescent tags. And then in the 80s, biologists were able to record the changing architecture of the cytoskeleton. So literally, you see this happening over the course of like 40, hit the wrong button, cut myself off. So over 40 years, as you can see that it's changing and happening, where we're developing this idea just to see what's happening in your muscles. Like, don't take that for granted. When I can tell you about what's going on with actin and myosin and how they're able to kind of line up and they attach to each other and move out, like, if you get to learn that one of these days, don't take that stuff for granted, because people were researching for decades to figure that stuff out. Just insanity, which is awesome, because then we get to know that. We get to feed our brains with it. So I see that we're at the 10-minute mark. This would be a great place for you guys to take a break, take a little brain break, get up and stretch if you need to. If you really, really struggle and have to, you can go to the bathroom at this point. But do your thing and then come back and let's keep chatting. Okay, so the short, numerous appendages that propel protists, like paramecium, he's a real cute little dude, are called cilia. So they're just little hairs. So some cells you'll see they have little hairs. You'll see a better picture. But you might have seen paramecium before. Other protists move with flagella, which are longer and usually limited to about one or maybe a few per cell. Like E. coli is kind of this shaped, and it has 3. Okay, so some cells, multicellular organisms also have cilia and flagella. So you have sort of cilia through your lungs and through your respiratory tract to help move things along. Flagella, not all that often, though. We'll talk about that. But there's cilia, just some hair-like structures. That might be the best way to do it, is they're just little small hair-like structures. I guess I don't have paramecium here, but if you see it, it's kind of a this-shaped guy, and it's just full of those little hairs. Okay, so that's worth looking at. A flagellum, which is longer than a cilia, propels a cell by undulating. Undulating means it's whipping. So an undulation is just a whip motion. Cilia work more like the oars of a boat, but they kind of do this weird undulation too. So just that whip-like motion. Okay. Although differences exist, flagella and cilia have common structure and a mechanism for movement. So they're both still kind of doing a whip-like thing, just to get movement to happen. And they're all going to be made of sort of the same cytoskeleton, by the way. Okay. Let's see. So flagellum is common. The only real animal one, or at least human one for sure, is sperm. It has the long tail of the flagella to allow movement. Okay, so both flagella and cilia are composed of microtubules that are wrapped in an extension of the plasma membrane. So the whole idea here is that it's just cytoskeleton that comes kind of out of that plasma membrane. There's a whole thing that we could go, there's like a 9:2 ratio or whatever. We're not going to get into all that. If we know that basically it's cytoskeleton and microtubules that extend from the membrane, then we're fine. That's all I'm asking you guys to know. Okay, so there's this whole like 9:2 arrangement, but the idea is just all of these complex structures in here can really be broken down to the fact that it's still a part of the cytoskeleton. It's still a part of that. And I mean, a good way to think of this is like, if you guys have some pets at home, it's like an animal's tail. It still has bones because it's an extension of the vertebrae. It's an extension there. So it continues to have the bone structure here. I'll draw you a puppy. You can have a puppy today. And put a little nose on him. So the tail is still actually part of that whole like vertebrae. And that's the same here. So it's still part of its skeleton. The tail here, or even some of those hairs, are still part of the cell's skeleton. So it's still similar at that level. Let me know if that's like way off, then delete it from your brain. But if you can sort of see it, I hope that helps make a connection. Next thing I want to talk about is that animal cells synthesize and secrete what's called an extracellular matrix. So extra is just going to be outside of the cell and matrix. So it's just a whole, it's a network of stuff. The matrix is kind of like a network of stuff that's outside of the cell. Okay, and that extracellular matrix actually holds cells together in tissues and then protects and supports the plasma membrane. So the ECM is actually pretty important. And you can sort of see it in a picture. Actually, I don't want to worry too much about integrins. There are some integral proteins, is what we call them, but proteins within the membrane called integrins that hold the ECM, basically. It's as much as we'll do on that one. But this is sort of what you can see. This is much better to look at. So you can see the membrane. See those hydrophobic, sorry, let me get my hydros in the right area. Hydrophilic heads towards the outside, and those hydrophobic tails towards the inside pointing to each other. Okay? But within those, we'll also have some proteins. And if you see, they're pretty large proteins. They're called integrins because they're integrated into the entire membrane. You have to look at integrated. It just means like put together with the entire membrane, right? And so they serve as an attachment for all this stuff going on the outside. Notice it's just a matrix, a whole bunch of connecting tubes that's outside of the cell. So that's the extracellular matrix. Do I want you to know collagen fiber and connecting and glycoprotein? No. Just know that there's a part outside of it, outside of the cell, that has all of these networks to help hold everything in place. Okay? Really quickly, we'll go into cell junctions, just so you're aware of them. I don't typically ask too many questions on this one, but it's nice to see these, particularly if we're going to do anatomy in the future. They love to start with this. So you can't have a tissue without all these junctions. Okay? So adjacent cells will actually adhere, interact, and communicate. Your cells have to communicate. If you damage one, it's got to talk to the others and say, we need to fill this space. You need your cells to communicate. That's how your heart keeps beating together, is that it's communicating with each other, all those individual cells. So we have some tight junctions that prevent leakage of fluid across a layer of epithelial cells. Epithelial is just like a lining. Okay, so tight junctions will help kind of make sure that we don't have leakage in the lining. So think like skin or your like intestines may be lined with stuff. You don't want leakage out of your intestines, I promise you that. Nope. So we need prevention of leakage in the lining. Anchoring junctions help fasten cells together in sheets, so we can sort of get some tissues in particular sheets. We'll have gap junctions, which are channels that allow small molecules to flow through in these protein-lined pores between cells. So these are really the communication lines. So communication happens through the gap junctions, through the little holes in it, because that's where we're going to get stuff back and forth. Okay, so you can sort of see what they look like. This is a tight junction. It's a lot of, I'm thinking if you've ever sewn, it's sort of like when you use a serger and put things together. There's lots of lines here. If you haven't, just think of, it's like sewing a whole bunch of stuff together. So these things are very tight. We do not want to have leakage between cells. Anchoring junctions have a lot of extra bits. Again, it's just to kind of keep things anchored and keep it holding on to what it needs to. And then gap junctions allow for communication all the way through. Okay, just so you can see those. All right, so a plant cell, not an animal cell, has that cell wall. This is super important. So plant cells with a cell wall. This actually protects and provides skeletal support that helps keep the plant upright. It's primarily made of cellulose. Can you figure out what that is? I bet you know. So because they're surrounded by this tough protecting cell wall, they also still need to communicate cell to cell. So instead of having a gap junction, they're going to have a plasmodesmata. Big fancy word that just says a hole is poked in the cell wall. So a hole in the cell wall. Because how would you get anything through there if you don't have a hole in it? I mean, come on. So through it, they'll get water and nourishment and chemical messages. All right, so you can see that there, it would be sort of a gap, as it were, but because we have the cell wall here, it also needs a break in the cell wall so we can get things through it. Okay? So just the holes in the cell wall are called plasmodesmata. There you go. Quick review. Eukaryotic cells can be grouped based on 4 functions: genetic control, manufacturing, distribution, breakdown of materials, energy processing, structural support, movement, and intercellular communication. We covered these 2 today. These were all those, I lost the term for it, endomembrane, and this is basically just nucleus stuff. So take a look at that. The whole point, and we're going to go through the chart more together now that we've completed it. So I wanted you to have the chart, to look at it. We'll make sure that it's completed in the next class period. But again, these are some ones to look at as far as what they are, where you find them, and what their purpose is. So you have access to this in the live binder. If I need to make it bigger, I can, but to know it, I encourage you to write it yourself. You got to do it that way. Okay, we got our energy processing, again, pictures. We've got the structural movements there. And again, these are probably good to know. Extracellular matrix, basically what cell junctions do, the cell wall. Like, I like this list. This list makes me happy. Hint, hint, wink, wink. That's why you have a table. If you didn't catch that. All right. Okay, so this is where we kind of finished. We're finished with this chapter. We're going to go through and we're going to do a little bit more with it, check with cells. We're going to make sure we review it because it is a hugely important section. Very, very important. This also means that if you have any questions, you got to ask them. Because every year, everybody makes it seem like, oh yeah, we got this, we totally got it. And then we get to the STAR test and we don't remember any of the cell parts anymore. So I see it every year. So I'm just saying, the more we can put in that work and effort to know it now, the less work down the road. Because I'm telling you, this is a chapter that the STAR end of course, they love it. Oh, they love it. A good way to help you review before I put one up too is that I'm leaving this. I don't have it on here too much, but it will definitely be in the lecture notes section. It gives you all the you should be able to's from the whole chapter, and there's quite a few. Come on, marker, work for me. But there's a lot here, so check through these. And if one looks particularly tough for you, like I don't know what this means, just ask. Okay, but again, I'm not going to harp on that too much because you'll have it for you. It just is an extra resource for you guys to use to make sure that you get this section, because I'm telling you, it is important. All right. Again, any questions at all, whenever they come up, feel free to shoot me an email, and I promise to get back to you guys.